In this video, I'd like to be the first in a series. I'd like to begin to consider the immunohistopathology on the glioblastoma specimens that were sent to me. Let me get my pointer fixed here, sorry. On this first slide, I want to present two main ideas. One is first that it's the primary tumor that we're performing the histology on. It's the immediately adjacent area, that in the proximity of the portion that is removed, that typically will be the source of the cells that are that terminate the patient, if you will. So that's the trouble zone, but we know that they spread beyond there, and it's all going to be staged. If we beat the perimeter, then these will become the problems. This entire panel on the right hand side is from this study in the Archives of Neurology, volume 69, number 4, April 2012. And they used an antibody staining the mutated, it's the HO9 antibody, I guess, against the mutated IDH1 enzyme. It's not perfect, but it's very interesting. First off, that they had whole brain paraffin sections to be able to stain on this. But anyway, here's some of the staining. It just reminds us of what we're dealing with and what we're focusing on. Over here on the left, we're thinking of how these cells might spread. And in the top left, this is from work where I think this was actually in vivo imaging, and they would put in the C6 GBM cells, I believe what they are. They seem to go into the rats and mice quite nicely under certain conditions. But anyway, this is what they observed. There's migration along the vasculature. So you notice that the vasculature is growing into this site, or this is existing vasculature, and it's beginning to spread along it to grow into a larger mass, however you want to look at it. And then there's a certain point at which a critical mass is reached, and that spread along the blood vessel is then joined by the second type of cell. They called it the multipodo cell, with all these extensions coming off it, and then they spread away. It's very characteristic. They'll send these tentacles out as feelers. They'll be pushing out and retracting, and then some will push and stay there, and then the cell will migrate in that direction. A lot of dynamics, wonderful paper to read, and just really inspirational that someone would do this much work. Here on the bottom left is work from slices, the tissue culture. So there's no blood flow through these vessels, and I think this was it's in vivo. There it says right there. So there was blood flow through these. So the conclusion drawn by the second group is that the attraction of the glioma cells to the vasculature does not depend upon the content in the vessels. So it seemed like a good argument. But they observed the movement and the progression and division along the way. Sometimes after they divide, one daughter cell would go one way, they go another way. A lot of very interesting observations. This is an interesting one down here, that the average speed of cells that divided was 23 micrometers per hour. And it wasn't very different from those that didn't divide during that, 22.6. So do all these spread through vasculature? Of course, I think since the 1940s, it's been claimed that it's both along the vasculature and within the myelinated tracts. So the challenge I'm going to place in front of you is, how do they actually do it? What's the environment that they're within? It's solid tissue, as you can see here. There's no empty space whatsoever. So how are they working their way through there? This is the way that the oligos will move. and they'll like to move along an axon. So here's a glial cell, and this might have been in vitro where they actually watch the oligos move along. So we're picturing oligos here. Of course, it's showing them already forming the myelin in the CNS, but do they migrate along? How do they, if they are passing through a tract, how are they working their way through all of this? What are they actually attaching to? And supposedly, in insects and other organisms in the periphery, there will be this tip cell that will start, and there'll be follower cells. Well, if you read through these articles, they talk about how one will proceed and then it'll draw the others along with it. So this is a very good hypothetical model. Again, we're working with tumor cells, so why should they behave and only migrate as single cells, as claimed by the authors in this Nature review? And as these oligo precursors are going to migrate, what form are they taking along the progression? Of course, there's tremendous amounts of signaling, and here, of course, this is in the P30, so this is the developing mouse brain, I guess they're looking at here. They certainly look like they're all lined up as they are here, which I guess they would be if they were forming the myelin like that. How are they hanging on to the axons themselves? Supposedly in this bottom left there's some communication, PDGF involved, neurotransmitters, depolarization of the oligodendrocyte progenitor cells, JAG get involved, ATP adenosine, you get the myelin gene promoters affected, and so a wide variety of events occurring for the differentiation. So if they don't differentiate, do they keep moving? Can they move as far as we think, just along the axonal fibers? In the myelinated pathways, can they get to the axonal fibers? So let's look at some electron microscopy. These two here are actually the optic nerves, but they're CNS, and this is a peripheral nerve. So in the optic nerve, I think this is the bumblebee, actually, whatever it is, and this is the salamander, but it doesn't really matter. We have the astrocytes, the oligos, and microglia. That's an endothelial cell up here in the top left. And we see all of the axons in cross-section here, some in longitudinal or oblique, and the oligos wrapping around all of these, multiple wraps, as we would see over here, large and small. And any other space, so as we see here on the illustration, or here with the green being a single oligo and then the axons in red, there's no empty space in the brain. It's all the rest of the space is filled by astrocytes. We don't have the case like in the peripheral nervous system and the nerves, where we have the endoneurium, collagen, the classical types of connective tissue, the extracellular matrix that you might be thinking about. Not quite so classic, special for peripheral nerves, but this also is reminding us that here's a bundle of axons that are surrounded by an oligo, a Schwann cell in the PNS, but an oligo in the CNS for unmyelinated axons, right? So this is not a myelinated axon here. They're not seeming to illustrate it there. So are they unmyelinated fibers that the cells are grabbing onto and migrating away from the primary tumor? So here on the bottom right, this is an oligo. It's actually a mature oligo in here, but we're trying to picture an early oligo. How is it moving through the white matter to well, I'll call it metastasize, but anyway, it's staying within the same tissue, but it's just moving to a new location. So what are the barriers? What does it have to overcome to move through this part of the nervous system? That looks a little bit like connective tissue there, but again, this is the optic nerve, still it's CNS. So if it is not moving along a bare axon, or squeezing in between the, still it's got to squeeze in between single wrapping of the oligo and the unmyelinated axon, then it's riding along the surface of the oligo that's already there. So how does one cell move on the surface of another cell, whether it's the axon itself or another oligo? Might not be over the somal part of it, where the cell body is. It might be out on some projection of that. So there's a cell junction that's involved, and then, as we'll see, we'll focus more. This is where the vimentin, the nestin and the vimentin become important in thinking about the inner workings of the cell, of how to squeeze out a leading edge, attach, and then pull all the guts up to that point. That's our nestin and vimentin are going to, as well as many other molecules, of course, contribute to that. But these are the things we have to think about, what's going on. Well, let's start first with the first result here. Synaptophysin is a neuronal marker. All that I see in this are axonal swellings. These are beaded axons, all of these positively stained structures here. The general brownish coloration I'm assuming to be background, but all these little punctate structures are characteristic. First thing I thought of when I saw it was axons. I've stained for a lot of peptide transmitters in the rat brain and human brain and baboon brain, and this is what they look like. So is this what's happening in this area? Here in the site 2, I don't see as much of that, so this is where I think the vascular effect is occurring, and I'll tell you about that and more about that as we proceed. And sorry for the hesitation there. So let's think about the vasculature in the white matter and the gray matter. So we know that the vasculature to the CNS comes from the periphery toward the center. So here's the gray matter in the cortex and the white matter in the deeper part. How does this vasculature change as it penetrates down in? Of course, this is very well known. There's nerves that accompany the arteries in for a certain distance, and then eventually you have a change to where you've got the typical type of blood-brain barrier. So the endothelial cell with its basement membrane, and then smooth muscle cell, perivascular nerves for a PL artery, and it is going to become an arteriole as it goes in deeper, and eventually it's going to be surrounded by less and less smooth muscle. As we'll see, pericytes will replace those, and then the astrocytic foot processes will come into play later on. So what's the difference between the perivascular space? Basically, we're thinking about the basement membrane created by the adjacent cells, the endothelial cells, and whatever is adjacent to it. They're both contributing to that. So that perivascular space is very different for the vessels in white matter as it is in the gray matter. You can see very quickly in the two electron micrographs here, comparison of the perivascular astrocytic structure. So they do differ. You can see just kind of a difference in the appearance of the blood vessels. So where in here are these cells migrating? Are they migrating in between the myelinated axons, or are they working their way in between the astrocytes and the endothelial cells to get into the basement membrane there? So this is a slide for a totally different purpose, but in the last few seconds of this first recording, I just want to point out the sympathetic peripheral nervous system. But if we're thinking in the CNS, this would be an unmyelinated axon, a myelinated axon, and a blood vessel. So, here we're seeing the periphery, the Schwann cells migrating, but there's always migrating cells, peripheral, central. What are they on? What resolution do we have to be able to determine that? So we'll stop here with the first recording, and we'll pick up with the second one to build on these ideas.